Hello students, welcome back to my video. I'm Ms. Rukmanisha, your biology teacher, and we'll be continuing with chapter 11 where we were learning genetic engineering. So in genetic engineering, as we have seen in our previous video section, that is in our second video section, the steps which are involved in this genetic engineering, which we also known as recombinant DNA technology. Why? Because the first step of genetic engineering is to create recombinant DNA, right? In short, we call recombinant DNA as rDNA. And then the next step comes as gene cloning and gene transferring, and the third step comes as the upstreaming and downstreaming process. Okay, so the very first step of this genetic engineering is the creation of recombinant DNA. And to create a recombinant DNA, we require certain tools for this process. So yesterday in our previous video section, the 5 tools which we have written, okay, out of those 5 tools, one of the most important tool is restriction enzyme. And in our previous video section, we have seen another important tool, that is plasmid, which is known as a vector. The nature of the plasmid and the structure of the plasmid, how it works, that we have seen in our previous video section. In today's video section, we'll be seeing the last tools of restriction of genetic engineering, that is restriction enzyme. Okay, and this enzyme was discovered by, if you can recall from the biography of the chapter, it was discovered by Robert Bayer, right? Robert Bayer. Now, in our previous 2 videos, we have learned several things about recombinant DNA technology. Now, in this video, we will be learning the restriction enzyme, which is the most important part of the biotechnology. Okay, so this restriction enzyme, it is also known as molecular scissors. Molecular scissors. Why? Because they can make a cut in the DNA. If you can recall, that is what I was saying in our previous 2 videos. If you can recall, for genetic engineering, cutting the genetic material, that is cutting the DNA, becomes a very important part of the process. We need to cut the DNA. But how do we cut the DNA which we cannot see with our naked eyes? So we have to add this enzyme, which we call restriction enzyme, so that this enzyme can make a cut in the DNA. Okay, so this molecular scissor, why it is named as such? Because of its nature. Okay, restriction enzyme, they can make a cut in the nucleotide of the DNA, and that is why they are known as molecular scissor. And because of this restriction enzyme only, recombinant DNA technology has become possible, right? Because we need to make a cut in a DNA so that we can insert a foreign DNA in it, right? Without cutting, we cannot insert this foreign DNA in it. So if you don't insert this foreign DNA, recombinant DNA cannot be formed, right? To form recombinant DNA, we should make a cut here, and then we insert this desired DNA here. And that desired DNA also has come after the cutting only. So it must have come from some other DNA, from where from this portion it must have come. So we have made a cut here. So that is why this desired DNA also came from by the help of restriction enzyme. Okay. So there are 2 types of restriction enzyme. Excuse me. There are 2 types of restriction enzyme. One is restriction endonuclease and one is restriction exonuclease. Restriction endonuclease and restriction exonuclease. Okay, so the role of this exonuclease and endonuclease are different. Okay, so here the DNA makes a cut in on the basis of their cutting. Okay, they make a cut in the DNA in a different pattern. So on the basis of that cutting, their name is given as endonucleus and exonucleus. So endonucleus are those nucleus which cuts within the DNA. So this is DNA. Within the DNA means in between the DNA here. Suppose the cut is made here. Suppose the cut is made here. Okay. So that type of restriction enzyme is known as restriction endonuclease. And restriction exonuclease means the DNA when it is cut from the end of the DNA. Okay. The DNA is cut from the edges. Okay, when you make the cut at the edges, then it is known as restriction exonuclease. Alright? So there are two types of restriction enzyme. The restriction enzyme which makes a cut within the DNA is known as endonuclease, and the one which makes a cut in the edge of the DNA is known as exonuclease. Next we will see the example of restriction enzymes. Okay, or these examples are such as HIND2 and ECOR1. These are example of restriction enzyme. Now, this restriction enzyme, how are they named? Their nomenclature. Remember, the restriction enzymes are extracted from the bacteria. Okay, they have been extracted from the bacteria, and there are around 200 different types of restriction enzymes which are removed, which are extracted from the bacteria. Okay, and different restriction enzyme makes a cut in a different pattern, in a different location of the DNA. Okay, there are more than 200 restriction enzymes we know, and all these restriction enzymes can make a cut in different location. Okay, because they are enzyme, and if you remember from class 11, that enzymes are quite specific in nature, right? Quite specific in nature means if they make a cut, these enzymes make a cut, they will make a cut in this specific location, specific site. Okay, and if this enzymes make a cut, it will make a cut in a specific location. It will not randomly cut in any part of the DNA. Okay, so let us see the nomenclature, why these restriction enzymes are named in such a way, and what are the rules these restriction enzymes are named in this manner. So this HIND2 is a restriction enzyme. As I have told you, restriction enzymes are extracted from bacteria. They have been extracted from bacteria. So different types of bacteria from where the restriction enzymes are taken out. Okay, to make a cut in any DNAs. Okay, these enzymes which are taken out from the bacteria, they can make a cut in their DNA also, in the bacterial DNA also, and in other DNAs also, such as in animal cells DNA or in plant cells DNA. So this restriction enzyme is taken out from Haemophilus influenzae bacteria. Haemophilus influenzae. Okay. Now, while naming this restriction enzyme, the first letter of the genus and the second two letter of the species, the first two letter of the species is taken here. You can see HIN. So H is from the genus and IN is from the species. And D refers to the strain. Bacteria can be of different strain. They can be of the same species. Bacteria can be of same species, but they can possess different strain. They function differently. Okay, they may be of same species, but they function differently. So that's when we give them as a name as a strain. Okay, different strain. So this bacteria has a strain. From D strain, this restriction enzyme has been taken out. So this is influenza, Haemophilus influenzae is of D strain. And the last Roman number named as Hind1, Hind3, Hind2. So this letter, this Roman number refers to the order of isolation. That means when the scientists were discovering this restriction enzyme, which restriction enzyme was first discovered in Haemophilus influenzae. From Haemophilus influenzae, we have extracted this restriction enzyme first, then this restriction enzyme, then this restriction enzyme. So according to the order of isolation, which one extracted, which one was discovered first, according to that, this last Roman number is allotted, okay, in the name. In the same manner, E. coli, we say it as E. coli, short, to make it short, we say it as E. coli, but actually it is not E. coli, it is E. coli. So we say it as Hind2, just to make it short, but it is actually Hind2, okay, Hind2. So nomenclature of E. coli is from, so you understand that this E comes from the genus, and the second two letters comes from this species. So Escherichia coli bacteria. Okay, from the bacteria Escherichia coli. Okay, from this bacteria, this restriction enzyme has been extracted, and this E. coli belongs to R strain, and the order of isolation is 1. That means this enzyme was first discovered from this bacteria. Okay, E. coli 1. Next. As we were discussing that in a DNA, it can be any DNA, a linear DNA, a chromosomal DNA, or a circular DNA. The circular DNA examples we were seeing, plasmid is a circular DNA, right? So this is a chromosomal DNA and this is a plasmid circular DNA. And what this restriction enzyme can do? This can do to this DNA, it can make a cut in this DNA. Okay? So when this restriction enzyme is making a cut, they make a cut in particular sequence. Where they make a cut, that region is known as recognition site. Okay, wherever they make a cut, this location, this region is known as recognition site. Okay, wherever they make a cut. So every enzyme have their own recognition site. Okay, they have their own recognition site. Now you know that DNA is made up of base pairs, right? A, T, G, C. Okay, the sequencing of these base pairs are in different patterns and random patterns, right? So here, if I talk about this location, obviously here base pairs will be there. Here base pairs will be there. Here base pairs will be there. Here base pairs will be there, right? Wherever the locations is. So what is actually the recognition site? Recognition site is a small DNA sequence fragment, right? It's a small DNA sequence fragment where actually this restriction enzymes can make a cut. Okay, where they can make a cut. So every restriction enzyme, we have seen only two examples, Hind2 and E. coli. That means E. coli we have seen, right? So there are actually many. So knowing the nature of one restriction enzyme is enough, right? Knowing the nature of one restriction enzyme is enough. So we will take an example of E. coli. Okay, so this is a restriction enzyme, suppose, and this restriction enzyme will make a cut here. Okay, so this location is known as what? Recognition site. And in this location, the sequence of the DNA, okay, the sequence of the DNA will be GAATTC, CTTAAG. So this is the sequence. So GAATCC is from 5' to 3' and CTTAAG is from 3' to 5'. So you know the polarity of the DNA, right? So 5' to 3', whatever the sequence is, so 3' to 5', it will be complementary, right? So complementary should be always G against C, A against T, always in DNA. So this is the recognition site of E. coli. So and every restriction enzyme has their own recognition site as this. Now you can see there is one particular nature of this sequence. Okay, 5' to 3', the reading of the DNA sequence is same for the opposite strand from the 5' to 3', GAATTC. So if you read it to the opposite strand from 5', it is GAATTC. So this nature of the sequence is known as palindromic. Palindromic sequence, or we call it as palindrome. The nature is known as palindrome. The sequence are palindrome when we read them from 5' to 3', and in the opposite end, the 5' to 3', the reading is same. So that is called palindrome. Okay, so the nature of this recognition site, where the restriction enzymes make a cut, that part of the DNA has a sequence, and that sequence is a very small sequence, okay, not a big sequence. It's a very small sequence, and that sequence is known as palindrome in nature, okay, because of the reading pattern. And all the palindromic sequence are like this. You can do one example by yourself. You can create your own palindromic sequence. Okay, you can create your own palindromic sequence. So this is the nature of the recognition site. Recognition site means, suppose this is a DNA, right? 5' to 3', and 3' to 5' here, and here is the sequence present here. So in this sequence is palindromic in nature. Why? Because they are reading, the opposite reading of the strand is same. If I draw any sequence here, suppose T, A, T, T, G, okay. Now its opposite sequence will be C, A, A, T. Now are they reading same? This is from 5' to 3', the reading is A, T, T, G. The opposite strand reading from 5' to 3' is C, A, A, T. A, T, T, G, C, A, A, T. It is not same, right? So this is not palindromic. Okay, the rest of the DNA, whatever the sequence is present, they are not palindromic. But yes, in the recognition site, in the recognition sites, the DNA sequence are palindromic. Okay, this is the recognition site where the restriction enzyme will make a cut here. Alright. Now, how do they make a cut? So that is another pattern which we'll be seeing. So this restriction enzyme ECO R1, how do they make a cut in their palindromic sequence? We will see that. So suppose this is the DNA strand. Suppose consider it as a plasmid or a vector. Plasmid, which we call it as a vector. So vector has a for this restriction enzyme, vector has a recognition site GAA TTC. GAA TTCG. So that is plasmid. Now we take a foreign DNA or a host DNA. Host DNA. A foreign DNA we will take, because while we are using the same, while we are using the restriction enzyme for the plasmid vector DNA and for the foreign DNA or the desired DNA, restriction enzyme we should be using same, right? Why you should be using same? Let us see here. So if we are using this restriction enzyme here, so obviously they will also have the same palindromic sequence. Same palindromic sequence this DNA also will have. Okay. Now you take this enzyme and how this enzyme will make a cut here. So this enzyme will, when you add E. coli here, so E. coli will break the bond here, phosphodiester bond, and it will break the hydrogen bond here. Okay, so it will break the hydrogen bond. Here, and the last phosphodiester bond here. So in this manner they will make a cut. So between the first two bases of 5' to 3' end, and between the last two bases of 3' to 5' end, so they will make a cut here. So in the same manner here also it will make a cut here. So this is 5' to 3', and this is 3' to 5'. Okay, so what joins G and A? Phosphodiester bond. What joins here G and A? All the nucleotides are joined by phosphodiester, and how the bases are joined by hydrogen bond here. So here it breaks the phosphodiester bond here, it breaks all this hydrogen bond, and it breaks the phosphodiester bond here. So in this pattern it makes a cut. Okay, so when it makes a cut like this, so we remove this part. Because the cut is done, so we have removed this. So after the cut is formed and we remove this extra strand from after cutting, the extra strand is removed. What do you see here? You see the hydrogen bond, free hydrogen bond here, free hydrogen bond. So this free hydrogen bond, they are able to attach with the other bonds, right? So that's why they are known as sticky ends. They will be able to form a bond with the nearest nucleotide. Okay, so what we will do, we'll bring this together, and can we join it or not? Yes, we can join it. So this process we are doing, but not we can't see this process with our naked eyes. Of course, this all process are done by the help of test tube and the enzyme and pouring. All this process we will be seeing in detail in our next segment. Okay, so here you see how it happens when we add enzyme and when we bring plasmid, or when you bring foreign DNA, how it happens. That is what we are seeing here. So now you with the help of DNA ligase, remember DNA ligase? Yeah, it is the tool of recombinant DNA technology. So with the help of DNA ligase, so DNA ligase will help these two sticky ends to join. So here was the plasmids, the vector DNA. So it will join. So this was the foreign DNA, which is now joined with the vector DNA. So what is this? Now they are known as recombinant DNA. So this is how we form recombinant DNA. So this figure you can see from your textbook 11.1. And 11.2 you will see a very rough diagram of the recombinant DNA technology process. Okay, so how we do this process which we have done now. So plasmids are circular, we know that, and this is the host DNA, right? Plasmid. Sorry, this is the foreign DNA. And what we are doing, we are trying to these are the steps, diagrammatical steps of recombinant DNA technology. Diagrammatically we are trying to show. Okay, so by the help of E. coli, example we are taking. It is not necessary that you always take E. coli, or we are supposed to take E. coli only. No, we can take so many restriction enzymes which are available. So by the help of this E. coli, we will make a cut here and here. Remember that whenever we take restriction enzyme, we use the same restriction enzyme for both the DNA. So this is a vector DNA. So we make a cut here. So we make a cut here. So what happens after the cutting? So this is our desired DNA, suppose. Okay, this part of the DNA is our desired foreign DNA. Okay, and here the plasmid gets open. Plasmid is also open. So with the help of DNA ligase, we will attach this fragment here. Right? We will attach this fragment here by the help of DNA ligase. And after that, we will insert this plasmid in the bacteria. We'll put back this plasmid in the bacteria. This is the foreign DNA which is here. Here's the foreign DNA. Now, when this bacteria will multiply, this recombinant DNA also will multiply. Right? That is known as amplification of the foreign DNA, amplification of the gene of interest or amplification of the foreign DNA. So here, when the bacteria is multiplying, it is making bacteria is multiplying into 2, dividing into 2. So from here, you can see the gene of interest, the foreign DNA here which is present, that is also becoming 2, from 1 to 2, right? So this process is known as gene cloning. Okay, this is called gene cloning because this is our desired gene. Cloning means what? Making a copy, exact copy, right? So this is the gene of interest, and we have transferred this gene of interest in the plasmid, and now we have transferred this plasmid in the bacteria, because plasmid can multiply only when it is inside the host cell, right? So plasmid cannot multiply inside outside the body, so they need to go to the host cell. So this is the host cell. So inside the host cell bacteria, we have transferred the plasmid, and as the bacteria multiplies, this plasmid also multiplies, and the gene of interest also increase in number with the exact structure. So it will not change its structure. So what the gene which we have transferred here, exactly the same gene will be formed here. Okay, so that is known as gene cloning. All right. So please go through this content thoroughly. If you have any doubt, let me know. In our next segment, we will be seeing gel electrophoresis and the selectable marker. Okay, thank you and have a good day.